Okay, welcome to chapter 7. This is photosynthesis, and basically it's the way that photosynthesis is operated and utilized to take light and convert it into food. All right, so let's start with the basics. Plants are photosynthetic autotrophs. Okay, now this word's important, autotrophs. So we'll talk more about that at some point. Okay, there's also bacteria that are photosynthetic, and there are organisms called protists that are photosynthetic. They're very, they're rare in a way, but they exist. Okay, so what we're looking at in these pictures, of course, these are obviously plants. That's pretty obvious. Okay, this is algae. Algae is a big source of photosynthesis for us. And this is cyanobacteria. Cyanobacteria is photosynthetic bacteria. All right, now, what does photosynthesis actually do? What is the purpose of it? So what plants and photosynthetic organisms do for us is they are able to take solar energy and convert it into chemical energy. And they release oxygen as a byproduct, which is something that we need to do what? To breathe. Right. So basically, to make it simple, they make for us food and oxygen, everything we need for survival. We could not live on this planet without them. That is the essence of it. Okay, autotrophs. So an autotroph is a self-feeding organism. To self-feed, okay, that's pretty straightforward. The fact is, they make their own food. They also make their own carbon source, and they make their own energy source. So they are completely self-reliant, I guess you could just say that. So they're self-feeders. Heterotrophs are other feeders, which means that they have to eat to get nutrients. So what's an example of a heterotroph? Okay, us. All animals. All animals are heterotrophic. Autotrophic. Okay, autotrophic organisms are things like plants that can perform photosynthesis. Some bacteria and some protists. Not all of them. Okay, but all plants for sure. All right, so we're here to learn photosynthesis, and to start everything off, we have to learn the anatomy of a leaf. So let's zoom in on the leaf. Okay, every time you see a picture, it's important to understand what you're looking at. Okay, see this right here? This little square and this big arrow. What does that mean? Okay, that means that you are now looking at this little tiny square of a leaf, and they have blown it up for you into this big image so you can see what's inside of the leaf. Okay, so let's see what's inside of a leaf. All right, now all leaves are going to have an epidermis, which is basically like a skin surface. So this is their skin right here, and then they have another layer over here, and in the middle, this is called the mesophyll. The whole middle section, that is what it's called. And inside, see these little weird kind of jelly bean looking organelles. Well, they're not really organelles, they're cells. They're called mesophyll cells. And see this square where it's opened up quite a bit? You can analyze a mesophyll cell. But before we do that, I want to make an important point. Okay, see these pores right here? This is a little pore. Okay, it is called the stoma or stomata. And yes, you have to know this anatomy. And what is the purpose of this? This is for gas exchange. What type of gas? Well, plants are the opposite of us. They inhale, well, they don't really inhale, but they bring in CO2 and they release O2. We do the opposite. We inhale oxygen and exhale CO2. So they're the opposite, so to speak. Now that gas is exchanged through the stomata. This stomata can open and it can also close, okay, at the plant's will. Let's go on. See this mesophyll cell? It's got a whole bunch of little tiny green things in it. Wow. Those little green things are the chloroplast, the site of photosynthesis. So pay attention to how they've boxed out one chloroplast and now they've given you this new diagram. Actually, I'm going to go to a different picture here. All right, the chloroplast. The anatomy of the chloroplast is incredibly significant. Now, there are only 3 organelles that have a double membrane, and the chloroplast is one of them. What's another one? Do you guys remember from maybe your cell chapter? Okay. Organelles with a double membrane. The double membrane is pretty significant. Now, there's only 3. Let's go ahead and list those off. The nucleus. Remember, the nucleus has a nuclear membrane. That's the double membrane. The mitochondria has a double membrane, and the chloroplast. Okay, and we discussed in previous chapters why that would be. Why did these guys have this double membrane? And if you remember correctly, it was because of the endosymbiotic theory. Okay, so the chloroplast, it has an inner and outer membrane. There's the inner membrane, okay, there's the outer membrane, and inside of it, it's got this kind of semi-solid fluid, it's kind of gelatinous, called the stroma. So all of this space, it's not air, it's filled with this gelatinous type fluid called the stroma. Now, do not mix up stroma with stoma, because that's a huge mistake. And unfortunately, that is the name of this. A lot of the times you'll see it called stoma, and a lot of the times you'll see it called stomata. Now, do not mix that up with this. And you do need to know the anatomy. The other thing here is, see this little stack, okay? And to me, they look like poker chips. Okay, so imagine that these are stacks of poker chips. Okay, the entire stack of poker chips would be called the granum. An individual poker chip is called a thylakoid. And notice this, if we cut open the granum, see this space in here? this dark space, this is called the thylakoid space. Now this is important because you need to know where you are in the cell or in the organelle at all times. Okay, the stoma or stomata can open and close. This is a microscopic image of an open stomata. This is a microscopic image of a closed stomata. So they can open and close as needed. Now, remember the overall equation of photosynthesis tells us quite a bit. Okay, for photosynthesis to occur, we need 6 carbon dioxide plus 6 water plus light energy. And what is made here? 1 glucose, and remember the formula for glucose, C6H12O6, okay, plus 6 oxygen gas. It is the exact opposite of the cellular respiration equation. Cellular respiration equation is 1 glucose plus 6 oxygen. You get 6 CO2, 6 water, plus 36 ATP. All right, so keep that in mind. And yes, you have to know this for your exam. This is exam material. Actually, everything I'm going over is exam material, but you need to know this formula. I would write it multiple times over and over. Usually in the classroom, you guys would definitely be writing this down over and over again. All right, now what's happening here is we're turning light energy into chemical energy, which is of course our glucose. Yeah. All right. Now, remember redox reactions from our previous chapter. So the same thing's happening here, except it's different. We're having a reduction-oxidation reaction similar to cellular respiration. And when we are analyzing what's becoming reduced, what do we look for? What does it mean to become reduced? Okay, so remember, reduction is the gain of hydrogen, and more specifically, it's electrons. Those hydrogens come with electrons. And we see that. We see 6 CO2. These 6 carbons in the carbon dioxide right here are the same 6 that are over here in glucose in the C6H12O6 complex. The exact same. So if I were to radioactively label these carbons and carbon dioxide and then allow a plant to perform photosynthesis and then analyze the glucose made, these 6 carbons over here would also be radioactively labeled. Okay, so that's just a fact. Now, water. Water has 12 hydrogens, this one in particular, because it's 6 H2O. And it eventually becomes 6 O2. So what has happened here? What's the point? It's become oxidized, and what does that mean? This is the loss of hydrogens, and again, more specifically, electrons. This is incredibly important. You guys, I want you to remember this, learn this, write this down. Even though you have these notes, it doesn't matter. You need to write it down because these formulas, the way you memorize them is basically by writing them over and over and over until you have them down and memorized. Okay, so memorize it. All right, so this is the road map, meaning the big picture to photosynthesis. And what's interesting about it is that it's very simplified. And then we're going to move to the next part where it's not so simple, and we're going to learn every single step, which is absolutely mandatory and necessary to complete your exam and move on. All right, so photosynthesis. It has two parts. The two primary parts are as follows. Let's see. Aha, okay. We have light. Oh, that's not working. Thought I had shapes. Aha, there it is. Okay, we have light reactions and we have dark reactions, aka the Calvin cycle. Now this roadmap, why is it important? Because it's a good reference point after we've learned all of the details. I want you to always go back to this roadmap and just put things into perspective. All right, so this is what's going on. We have a chloroplast molecule. Okay, they're specifically put in one thylakoid. Well, it's a little stack, but technically, you know, this is the thylakoid. And they left out everything else because of the fact that we don't need to look at it when we're dealing with these reactions. We need to look at the thylakoid because that's where the light reactions happen. And we need to look at the stroma because that's where the Calvin cycle happens, in the stroma. So when you're learning something like this, photosynthesis or cellular respiration, the first place you want to start is location inside of your organelle, location inside of the cell. So right now we're inside of a plant cell, we're technically inside of the chloroplast, and now we're going even deeper into the thylakoid and the stroma of it. Okay, so what do the light reactions require for them to occur? Okay, light reactions use two things. They use water and light. All right, now, is that everything in the formula? No, absolutely not. There's still 6CO2, but they are not utilized in this reaction. They are utilized here. Okay, now what do we get out of the light reactions? Big picture. We get oxygen comes out, and there's more. There's ATP. ATP is made. And look at this guy. He should look a little bit familiar. We get an electron carrier called NADPH. Now notice there's a P in there. Remember NADH as an electron carrier from cellular respiration? This guy's pretty similar. And a good way to kind of remember that, this is photosynthesis, is there's a P right here. So this is different than the NADH in cellular respiration because it's called NADPH. P for photosynthesis. Wow, not really, but it's a good way to remember it. All right, now these light reactions, they build ATP and NADPH so that they can drive the Calvin cycle. The Calvin cycle. The Calvin cycle is this very sophisticated cycle in which carbon dioxide enters the cycle, 6 of them, of course, and what is produced here is one glucose molecule, the C6H12O6. Now, in the meantime, it's going to use ATP and NADPH, therefore they're going to go back to that form of ADP and NAD plus an empty electron carrier. Okay, so before we jump into photosynthesis and all of the steps, we have to understand what we're talking about, what photosynthesis is, what solar energy is. Now, we know that all energy comes from the sun. Now, that energy can be measured on something called the electromagnetic spectrum, and it allows us to look at the different types of energy wavelengths and be able to measure the wavelengths, the intensity, and so on, and what type of energy is coming from the electromagnetic spectrum. Now we have different types. I'm going to zoom in on this picture and we're going to analyze it. Okay. Start over here with radio waves. Okay, radio waves are quite large. Of course, all of these are not visible except for visible light. Okay, radio waves are quite large. They tend to have low energy, but they are effective. Then we have microwaves. Microwaves are used in our microwaves themselves, our cell phones. They're pretty large waves, and this allows us to have the communication that we do have. And then we have infrared. Infrared is, of course, comes from the sun. We feel it as heat, but of course it's not visible. Then we have this little tiny sliver, okay, of visible light. Here it's amplified or pulled out so we can look at it, and we can actually measure the wavelength in nanometers. So notice the visible light. We have violet, blue, green, yellow, orange, red. Okay, and visible light is in this very small spectrum, meaning the waves themselves. And what I mean by wavelength is this. Everybody look up here. See these waves? The distance between this wave and this wave, and this wave and this wave, is called the wavelength. Now notice as we go this way, the waves get smaller. Now that's incredibly significant. All right, now visible light falls into the spectrum. If we measure the wavelengths, which we have done and can do, the visible light spectrum runs from 380 nanometers to 750 nanometers. Now after that, we have UV, X-ray, and gamma, and important factors about these. Now, visible light we can see with the human eye because we're capable of processing that. The rest of it we can't see. However, the shorter the wavelength, the more energy it contains. Therefore, gamma rays, for example. Gamma rays are tiny. They're tiny little waves that are jam-packed full of energy. And that's not necessarily a good thing for us, because what this can do is mutate our DNA and cause cancer. X-rays can do the same, and so can UV light. Overexposure to X-rays, overexposure to UV light, and that's the bottom line. Infrared, micro, radio waves, they're bigger, but they have less energy. So if you see these two arrows, increasing wavelength this way, and increasing energy as we get smaller. Okay, now why does this matter? Because that energy in visible light is harnessed by very specialized plants, plant absorbing photons. Okay, now this is a new word for everybody. I want you to learn this and understand. Oops, keep doing that. There we go. Okay, photon. Photons of energy. So inside of our plants, we have pigment. Now, we also have pigment in humans. Ours is melanin, right? And that's that. Now, plants have their own pigment, and that pigment carries photons. And photons are capable of absorbing the visible light waves that come in contact with it across the visible light spectrum. Now see the sign here? This means wavelength. And that's just a general sign in math, science, physics, etc. All right, so plants have photons, and we're going to learn, pigments, and we're going to learn 3. There are others, but we're going to learn 3. Okay. Chlorophyll A, chlorophyll B, and carotenoids. All right, now this is incredibly significant. Chlorophyll A can absorb violet and red light. All right, however, it reflects green and yellow. All right, therefore, this is why plants look green. Most of them, right? Okay, because of the fact that chlorophyll A reflects. Oh, goodness. Keep doing that. Sorry, guys, just bear with me. Because of the fact that chlorophyll A reflects green. That's why plants are green. Okay, now chlorophyll B is capable of absorbing blue and red. Okay, so it's a little different, but the fact is that it also reflects green and yellow, making plants green. Now what's interesting is carotenoids. These are a classification of pigments, and they can absorb violet and blue light, reflecting yellow and orange. So the term carrot, a carrot is what color? Orange, of course. That term comes from this pigment, carotenoids. Okay, so let's take a closer look at exactly how these pigment molecules are capable of absorbing photons. So the fact that visible light, it's coming through in wavelengths, aka photons of light. This is a lot of energy. High energy. Okay, and here we see a photon is hitting a chlorophyll molecule. It's not telling us exactly which one, but it doesn't matter at this moment. Now see what happens when the photon hits the chlorophyll. See this big arrow? What that's demonstrating is this excited state of this particular electron. Okay, electrons in general. So we know that electrons have energy. That's something that we've already talked about. Now they do, and I'll remind you here. And so you collaborate, you put these two together, photon plus electron, you're going to get an excited state, and that's what we have here. And this is part of the very interesting part of photosynthesis and how plants can harness solar energy. All right, so that's an important factor. Let's go ahead and move on. Now, this is something you could read through. This is basically, I've already explained these things, but this is just extra material for you guys. But this picture is significant. Oh, I didn't mean to do that. I meant to do this. All right, so this picture is, this is the chloroplast molecule. It's showing the thylakoid itself, which is carrying tons and tons and tons of pigment, chlorophyll, right? Because how do we know? That's obvious. It's green. If it's green, it has chlorophyll, right? Why is it green? It's absorbing all kinds of colors. How come it doesn't reflect, or I'm sorry, how come it doesn't turn red or blue? Okay, it's the simple fact of this, and this is a very important answer here. Okay, the reason why plants are green is because they absorb they absorb their visible light spectrum. Each one absorbs a different color, but the reason they're green is because they reflect green light, and we can see that. And we can see the other ones too, really, but at the end of the day, this is what makes plants green. All right, so that's that. Let's go ahead and look at the next significant topic. This is called the absorption spectrum, meaning that scientists have researched and researched and researched how these pigment molecules work. So how do we know that chlorophyll A absorbs violet and red? And how do we know that chlorophyll B absorbs blue and red as well? And how do we know? This is how we know. Because we have technology that allows us to harvest and isolate pigments like chlorophyll A, B, and carotenoids and run them through a machine and pull up a graph like this. Okay, and it tells us exactly what wavelengths, okay, of light are being absorbed percentage-wise from 0 to 100 on the y-axis, and then on the x-axis, what does it say? It's the wavelength in nanometers, from 400 nanometers to 750. And if you find chlorophyll A in green, you see it peak here and here. Notice that it doesn't peak here. That's because it doesn't absorb in this frame. Okay, chlorophyll B absorbs here, and more specifically this color, right? We could box that out if we wanted to. And then it also has some absorption here. Look at carotene. It only absorbs here. Past this frame, no absorption, therefore reflecting the orange and red. All right. So we looked at this roadmap earlier. It's a good reference point. Now let's go ahead and jump right in. In the light reactions, light is converted to chemical energy in the form of ATP and NADPH, the electron carrier. In the Calvin cycle, chemical energy from the light reactions, meaning the ATP and NADPH, are used along with CO2 to make sugar. Right, so let's see how it's done. First things first, inside of the thylakoid membrane. Now, remember, we're talking about if we're in the thylakoid, what are we dealing with? Are we dealing with the Calvin cycle or the light reactions? Okay. If we are in the thylakoid, it's the light reactions. Now, inside, embedded into the thylakoid membrane, plants have something called a photosystem. Okay, a photosystem is this big protein. Okay, and it's embedded into the membrane. Notice you can see this is the bilipid layer. Okay, and then not only is it a protein, it's a very specialized protein because it has pigment molecules. Pigment, 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 pigment. And then it also has these very, very, very important pair of chlorophyll A molecules. Important. Now notice this little box here. This is called the reaction center complex. This whole photosystem is a light harvesting complex. Now this doesn't exist anywhere else except inside of a chloroplast organelle, specifically inside the thylakoid. All right, so we know the anatomy, and let's see how this all works. Okay, the light reactions have two photosystems, an electron transport chain, and it also has ATP synthase. Now we've seen that before, right? Where have you seen ATP synthase? Question. Okay, think about it. And if you're thinking about the electron transport chain and the last step of cellular respiration, you are correct. If you are not, you need to review cellular respiration. All right, let's go ahead and dive in. Okay, this is a nice little clean setup. Now, again, you know what? Let me just be extra clear. First of all, every time you see a picture, what do? You break it down. We're inside of the chloroplast. More specifically, we're inside of the thylakoid membrane. Do you see that little tiny square? And see how it's blown up to show you the thylakoid membrane itself? And don't forget the thylakoid space, which would technically be here. All right, so here we go. Light reactions. This is how it works. First of all, we have photosystem 2 in line. And then we have an electron transport chain, and then we have photosystem 1. All right, notice really quick, and this is incredibly significant, that photosystem 2 comes first, and then photosystem 1 comes second. Okay, and the reason for that is because they discovered this photosystem first, and they called it photosystem 1. And later, they discovered photosystem 2 and realized its position was here. But they didn't change the numerical, they didn't change the numbers, they didn't change the name. So always remember that photosystem 2 in this chain, in the line, and in the process of the light reaction is first. All right, here we go. Now, light, photons of light are going to come in from, of course, there's only one place, the sun. Okay, and they are going to get picked up and absorbed by pigment molecules inside of photosystem 2. Pigment molecules will pass the energy, the photons of light, from one molecule to the next, And notice how it's going downwards. It's losing its momentum. It's losing its energy, right? And that's not going to work here. We need a big pow. So how do we get this, the accelerated energy or this excited energy? Well, of course we get it from electrons. There's only one place to really get it from. And it comes from, very specifically, this process. Notice this process carefully. What is happening here? Water is being split. Okay, water is H2O. And when it is split, it does something very special. It releases electrons. See these two little guys right here? Let's zoom in on them. These are electrons. And when water is split, they're going to be shot or passed to chlorophyll A and combined with the photons of light that are coming from the sun, allowing or creating an elevated state. See how this arrow jumps up? Well, it gets big and wide. It's showing you an excited state of these electrons, and they're going to go where? To the reaction center. All right, now this is an important factor, so take notes on all of it. But this process that I just discussed with you, the splitting of water, okay? Splitting of H2O is where we get our electrons. This is the source of electrons for photosynthesis. Okay, now, oops. How to make this a little smaller, guys? All right, there we go. Now, this splitting water is also referred to as photolysis, which is photolysis, the splitting of water. All right, so when you watch a video, when we watch our video later, you're going to hear them say photolysis. It's just a different term for the splitting of water. Okay, so let's recap really quick. Okay, so we have sunlight energy coming through, okay, from in the form of photons of light, which are picked up by pigment molecules and pass from one pigment molecule to the other, to the other, to the other. And they're losing their energy and momentum, so they get this boost of energy from the splitting of water and the collaboration of photons plus electrons, and electrons are in this heightened state and sent to the reaction center. Well, they can't sit there forever, so they're going to get passed through this electron transport chain, okay, by shuttles and proteins. Now, remember the electron transport chain we discussed before? Every time electrons pass through an electron transport chain, it's going to pump hydrogen ions across the membrane. In this case, they're getting pumped into the thylakoid space. Every time a set of electrons crosses this electron transport chain, that is what's going to happen. A hydrogen ion will get pumped across the thylakoid membrane and into the thylakoid space. Now, meanwhile, this happens, and these electrons, they get passed from the electron transport chain to photosystem 1. And remember, photosystem 1 is still a photosystem. Therefore, it's still absorbing photons of light, just like photosystem 2. And it's going to pass it from one pigment to the next, to the next, to the next, until it meets those electrons that came from photosystem 2, which initially came from water. And that's going to cause the same reaction. It's going to cause a spike in energy, and it's going to shoot these electrons up into the reaction center. Okay, great. Now what? Well, they can't sit there forever, right? So they're passed on. And who picks them up? This guy. NAD+, an empty electron carrier. And as soon as these NAD+ picks up these two electrons, is immediately converted into NADPH. And there's the electrons, right there and there. Done. That's it. At least for now, for this part. Where does that NADPH go? To the Calvin cycle, because it's going to help operate that thing. All right, now meanwhile, these water molecules are being split. And remember, the formula for cellular respiration is not one water molecule, there's 6 of them. So they're going to keep coming through, increasing the concentration of hydrogen ions inside of the thylakoid space. And as we remember from cellular respiration, the concentration of hydrogen ions going up in an area is going to lead to a big change. They can't sit there forever. So by the laws of diffusion, what's going to happen is they're going to move down their concentration gradient. And don't forget, they have an affinity for ATP synthase. And they're going to move down their concentration gradient through ATP synthase. And once they start flowing, they're going to activate ATP synthase and start churning out ATP. Now, it'll make one at a time. And that ATP is going to go where? One place only, the Calvin cycle. Okay. And those are the light reactions. Well, that is the light reaction itself in a nutshell. Alrighty, so let's see what's next. Okay, I'm going to recap. Okay, photosystem 2 absorbs photons of light from the sun. So that means it has to be daytime for this to happen. Okay, and it's going to, we're going to see that water, the splitting of water and the release of these electrons, and the collaboration of the electrons with photons is going to give us this high energy excited electron state. Okay, those electrons will be passed from photosystem 2 down the electron transport chain, causing hydrogen ions to be pumped across the thylakoid space, across the thylakoid membrane into the thylakoid space. And then those electrons will be passed where? To photosystem 2. Photosystem 2 is also capable of absorbing photons, and these electrons plus photons will clash or collaborate, and together they will lead to this elevated excited state of these high energy electrons, which must be passed on to something, and therefore it's passed on to an electron carrier, NAD+, which changes to NADPH, and that is sent to the Calvin cycle. Okay, meanwhile, because we have this high concentration, see all these hydrogen ions, that's a lot. Now, by the laws of diffusion, these guys are going to follow their concentration gradient, moving from a region of high concentration to a region of low concentration, and they're going to move through ATP synthase enzyme. And what does ATP synthase enzyme synthesize? Well, ATP, of course. The name says it all. ATP synthase. Now that ATP is going to be sent to the Calvin cycle, and that is that. So light-dependent reactions. Let's kind of break this down really quick. What are the reactants and products? Pretty straightforward. In order for light reactions to take place, we're going to need light, sunlight, plus water. Why do we need sunlight? Why do we need sunlight? Okay. What does the sunlight do for us or for the plants? Okay, the sunlight is going to deliver photons of light energy. What is the water for? Water is going to give us electrons. Okay, so we need that. Those are the reactants, technically, for light reactions. Okay, the products. ATP, NADPH, and then finally the byproduct, oxygen. So where does the oxygen come from that we breathe? There's only one place. It comes from water. All of the oxygen in our atmosphere that we depend on for life comes from this little tiny process called photolysis, or the splitting of water. Righty, and where do the light reactions take place? Inside of the thylakoid membranes. Okay, so things you want to know, you want to know all those steps. I suggest drawing the whole thing out. That's what we would normally do in class. And then you also want to look at the highlights, the reactants, products, and location. Very good. So what do these products do? ATP is ATP. It's going to take ATP to build sugar, which we have not done yet, by the way. Make sure you know exactly where we are in the reaction. No sugar has been made. We need ATP, which comes from the light reactions. We need NADPH. These electron carriers are going to carry electrons. Plus, they're carrying hydrogen, which is super important in building an organic compound. And of course, we're going to need that CO2, but that comes from the atmosphere. Okay, so far, the light reactions have given us everything we need to run the Calvin cycle. In other words, everything we need to run the Calvin cycle, ATP, NADPH. And there's more stuff, but right now, that's it. Now, oxygen is a byproduct of photosynthesis from the splitting of water and or water oxidation and or photolysis, but that's where our oxygen comes from as a byproduct. Okay. So that's a good recap, and then we're going to move into the Calvin cycle.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions.
All right, so the next thing we're going to talk about is the Calvin cycle, also referred to as light-independent reactions, meaning that they can occur without light. In other words, they can occur without sunlight. So no sunlight is required, unlike the light reactions. So in the Calvin cycle, essentially what's going to happen here is carbon dioxide, which is part of the formula for photosynthesis, is going to be utilized to make glucose. Now remember, carbon dioxide only has one carbon. This is an inorganic molecule. Glucose has 6 carbons and hydrogens, and it's very much organic. All right, so this is quite an amazing phenomenon. Only plants can do this. Only plants can take, photosynthetic organisms can take an inorganic compound and turn it into an organic compound. All right, so here we go. Important facts. This occurs in the stroma of the chloroplast. No sunlight is required. We've already established that. And the products that we have just acquired from the light reactions are utilized to run and operate the Calvin cycle. Okay, now there's 3 primary steps, but I break this whole thing down into 5 steps because in truth, that is what's happening. There's 5 steps to the Calvin cycle. You'll see them say 3 sometimes, and typically it's the first 3. Carbon fixation, the synthesis of a molecule called G3P, and the regeneration of ribulose bisphosphate, or RUBP. Okay, now these also occur, these steps. However, you will see at times either they will say that this is a step or they'll just shove it in there as material and expect you to know that it's happening. I like to teach it as a step so you know exactly what's happening all the time. All right, one molecule of G3P is going to leave the cycle. 2 molecules of G3P combine to make glucose. Now that's actually pretty significant. 2 molecules of a compound called G3P come together and make glucose. Now you've seen G3P before. Where have we seen G3P? Where? Anybody remember? If you have remembered, you should remember, we see this in glycolysis. Okay, we remember we go from, in glycolysis, the steps I taught you was glucose, okay, is broken down into temporarily G3P, which eventually becomes, really quick, this is 2G3P, just so we're incredibly clear, which becomes 2-pyruvate. Okay, so it shouldn't be too surprising that the reverse, meaning taking these 2G3Ps out of glycolysis, allows us to go back to glucose. Okay, either way, the fact is this. 2 molecules of G3P make glucose. G3P is a high energy 3-carbon compound, and it's very significant. Now, I'm going to teach you the Calvin cycle, and the way that I'm going to teach it is basically going to happen in this very particular way. It's the way the book teaches it. Okay, what's happening here is, remember, this formula has 6 CO2. Now, the people that wrote this book are highly intellectual, and instead of making this 1 CO2 molecule at a time, they're showing you 3 CO2 coming in at a time. Okay, now here's where this gets a little bit confusing, because when you watch a video, it's going to say that the Calvin cycle brings 1 CO2 in at a time, and the Calvin cycle runs 6 times. Technically, yes. However, we like to break things down and simplify them. So instead of running it 6 times, we're going to run it twice with 3 CO2 at a time. So everything is times 3. All right, so just keep that in the back of your head. Now, here we go. The Calvin cycle. The way it begins is, of course, CO2, our carbon source. See these little gray circles? What do they mean? Carbon. Okay, so CO2 has 1 carbon. It's a singular carbon compound. Okay, and for short, remember we call this a C. Oh, what just happened there? Okay. Something that has 1 carbon, we can call it a C1. Oh, okay, if this will let me type. I guess not. Let me try again. All right, hang tight, guys. Okay, carbon is a C1, meaning that it only has 1 carbon. Now this matters, this terminology matters, because when we're dealing with organic compounds, the number of carbons present is completely significant. All right, so the very first step is called carbon fixation. Carbon fixation is when carbon dioxide, this C1 compound, okay, is fixed to RUBP. RUBP is right here. How many carbons are in RUBP? Let's count them. 1, 2, 3, 4, 5. Okay, so that's fine. Now RUBP is a C what? C5 carbon compound. All right, now how do you fix something? How does carbon fixation happen? Like this. So what's going to happen is this. We have an enzyme, this guy right here, Rubisco. Rubisco is an enzyme. It's an enzyme that fixes, or we'll say connects, CO2 with RUBP. That's what it does. Now, this is an important enzyme. If I remove Rubisco, the Calvin cycle cannot operate at all. Okay, so the very first step is RUBP is connected or fixed to CO2. Okay, now think about those numbers. We're taking a C5 and adding 1 carbon, changing it into a C6. So they don't have it diagrammed here for you. So basically, I'm going to try to diagram it. Let's see. It's a C6. So 1, 2, 3, 4, 5, and 6. Okay, so this is happening in this area here, okay? Carbon fixation. Now, we have 1, 2, 3, 4, 5, 6 after carbon fixation. However, that's not going to work. That is an unstable compound, and it's going to immediately split into a C3. 3 here, 3 here. Okay, and that's carbon fixation essentially. And what do we get? We go from having 3 RUBPs adding on, which is a C5, adding on a C1. They break apart, and we get 6 of this molecule called PGA. Okay, great, PGA, fine, but that's not what we want. We want G3P. Okay, so how do we get it? Well, we use our resources from the light reaction. So we're going to have to invest energy, and there it is. We're going to go ahead and take 6. Okay, that's not working. We're going to take 6 ATP, and we're going to change this. We're going to change PGA into what we want, and we're going to need hydrogens to do that and rearrange this molecule. So we're going to take these electrons plus hydrogens from NADPH, and we are going to get 6 G3P. Okay. So that's looking good so far, so good. Now, what happens next is G3P, 1 of the 6. See how we have 6 of them? 1 is going to leave. It's going to leave the Calvin cycle. Okay, and that's just the way things are. And this kind of is just going to go off, and it could be used to build glucose. It could be used to build other compounds. For now, let's just say it's sitting here waiting for the rest of the cycle to occur. All right, so 1 G3P leaves, and we are left with 5 G3P. Okay, so this whole process right here is another step. Okay, Okay, so we have 5G3P, but we need to get back to RUBP because those 5G3P molecules need to be rearranged and we need to use them to continue the Calvin cycle. Now, something is not a cycle unless it begins and ends with the same exact product. If we don't do this next step, okay, which is the regeneration of RUBP, then the Calvin cycle would just stop. And that's not going to happen. That's not acceptable. So what happens next? The regeneration of RUBP. The cell is going to take these 5G3P molecules and it's going to rearrange and rearrange and rearrange all 5 of them until we finally regenerate RUBP. And of course, how do you do something like that? Well, you need ATP. And then we're back to square one. Okay, the cycle's going to run one more time. So if we're looking at the cycle in terms of the book, the Calvin cycle, it runs times 2. All right. So this whole thing's going to happen again. Okay, Rubisco will fix carbon dioxide 3 at a time to 3 RUBP molecules. They're going to immediately fall apart and break in half into a 3-carbon compound. So we're going to end up with 6-PGA. 6-PGA is going to be converted into 6G3P. And with the help of ATP and NADPH. Now we have 6, 1G3P is going to leave, okay, another one. So we end up with, in one photosynthesis cycle, we end up with 2 G3P molecules outside of the cycle, which is exactly what we need to make glucose and other compounds. Okay, and then we, of course, we go on. With the 5G3P left, we need to regenerate RUBP, so we do so and the cycle goes on and on and on. That is the Calvin cycle.
Plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Okay, so that's the overview of photosynthesis, and this is a good review to go back to. This is the photosynthesis roadmap, and now remember, the formula for photosynthesis is as follows. Okay. 6 CO2 plus, plus 6 H2O plus light energy and or solar energy. And what do we get? We get glucose, okay, which is a C6H12O6 organic compound plus 6O2, and that's the O2 that you breathe. Now, remember, the light reactions happen inside of the thylakoid membrane. And what's involved here, if you go back to your formula, only 2 things are involved. The water molecules, right? And the reactants and light. Okay, now CO2 has not been utilized yet. CO2 is not used at all until the Calvin cycle. Okay, now what comes out? What do the light reactions make for us? Okay, they're going to make the energy source to run the Calvin cycle, ATP plus NADPH, and it's going to make the 6-oxygen. Okay, now CO2 doesn't come in until the Calvin cycle. And even though we learned it as 3 CO2s coming in at once, technically that's the way we want to teach it, but it's 1 CO2 at a time. Ultimately, we get to G3P. 2 G3P will make our glucose. And this is a cycle, so it begins and ends with RUBP. Okay, so there we have, there's our glucose right there. All right, so we have identified the reactants, okay, and where they have a role. Light and water have their role in the light reactions. CO2 has its role in the Calvin cycle, and then our products, which is glucose and oxygen. Oxygen comes out of the light reactions from the splitting of water, and of course glucose is made by the Calvin cycle, and the Calvin cycle only. Okay, this is a good roadmap to review. This is the Calvin cycle once again, and I suggest that everybody diagram this over and over and over again and remember it all inside and out. Now, if we were in a face-to-face classroom, that's what we would do. We would be drawing this on the board, but obviously we're not there, so. Okay, now earlier we talked about the stoma, which is the opening, which is this little pore, this opening that basically allows gas exchange. And it's called the stoma. It's also referred to as the stomata. And that was early in the chapter. Now, of course, here we have an open stomata. Keep doing that by mistake. Okay. Open. This is a closed stomata. Now, why would a plant close its stomata? Well, it's going to do so if it's hot. If it's hot, if it's dry out and the plant doesn't want to lose water. All plants have this outer covering of wax that prevents water loss. However, if they close their stomata, well, the wax also prevents gas exchange. Gas can only be exchanged through an open stomata only. So what comes out? Oxygen, of course. Okay, and what goes in? CO2. This is called gas exchange because oxygen and CO2 swap. They exchange places. Okay, so if the stomata is closed, no gas exchange can occur, which means that oxygen can't leave and CO2 can't come in. That's a problem. Problem for plants anyway. Okay, so the stomata has to be open for this to occur, and this gas exchange is essential for photosynthesis and the survival of the plant. So what does this all mean? Okay, so a plant's going to close its stomata if it's hot, at the risk of not being able to exchange gas. So the plants will basically do it. And there are some consequences. So let's take a look at what those consequences are. Okay, so problems. Now, if the stomata is closed on a hot, dry day, the water stays in, which is a good thing. However, CO2 stays out, which is a bad thing, and oxygen, the byproduct, stays in, which is also bad. So why is it bad? All right, this is why it's bad. Now remember Rubisco. Rubisco is the enzyme in the Calvin cycle that fixes CO2 to RUBP. Okay, so normally that's how it goes. CO2, okay, I just had CO2 and Rubisco enzyme are basically interacting, and Rubisco fixes CO2 to RUBP, initiating the Calvin cycle, giving us sugar. That's good. That's what we want. Okay, but if we don't have CO2 in there and we have this, we have too much oxygen, that's an issue, a big issue. So what happens next? All right, so this is the issue. If there's low CO2 and high levels of oxygen, Rubisco will fix oxygen, which is not good because it's a complete waste of time and product. It's very bad for the cell. There's no sugar being produced. It's just wasting energy, wasting ATP, wasting NADPH, because guess what? The light reactions will still happen as long as the sun is out every single time. All right, so what happens when Rubisco binds to oxygen? That is called photorespiration. Okay, think of that term, photorespiration. All right, now plants that, when this occurs, this usually happens to plants that are called C3 plants. This is a C3 plant, it's a typical plant. Basically, let me just kind of break down some anatomy. Here's a stomata, also referred to as a stoma. Okay, these are the mesophyll cells all through here. Okay, these are all mesophyll cells. These little guys right here, that's the chloroplast. And notice this, it has veins. Okay, just like us, they need a vascular system to deliver everything. They need their nutrients. They also need oxygen waste taken away and CO2 delivered to all of their cells. Okay, now that's where the veins come in. They deliver this, and water, of course. Okay, now see how there's these bundle sheath cells surrounding the vein? Keep that in the back of your mind. All right, now C3 plants, they tend to go into photorespiration when it's hot, and it's very detrimental to the plant. However, over time, evolution has given us C4 plants. Okay, so a C4 plant, something like crabgrass, corn is a C4, daisies, and they have been able to overcome this. They're not susceptible to photorespiration because they have a specialized anatomy where they completely separate the light reactions. Okay, light reactions are happening here in the mesophyll cell. And they separate the light reactions from the Calvin cycle physically. So they have physically evolved this complete separation. So here's the issue. All right, if the stomata is closed and then the sun is out, the light reactions are happening. Okay, and what's the products of the light reactions aside from ATP and NADPH? Okay, oxygen. Get tons and tons and tons of oxygen, right? That's not necessarily a good thing because of the fact that it can bind with rubisco. But if they never can physically get in contact with each other, like in this situation, they can't. They can't touch each other. Oxygen cannot reach rubisco. It's not possible. So the point is because of this pretty brilliant anatomy, in my opinion, C4 plants do not have the problem of photorespiration. So what they do is this. They make sure they have plenty of CO2 to make plenty of sugar. So they bring in their CO2 and they're going to basically alter it slightly. And instead of just running it right into the Calvin cycle like a C3 plant, they convert it temporarily into a C4 molecule. Okay. Now that C4 molecule can move out of the mesophyll into the bundle sheath of the cell, and then it will drop off CO2 right into this Calvin cycle. CO2 is fixed by rubisco enzyme, run the Calvin cycle. And then it's converted to pyruvate, and it just goes on and on and on. But the point is that photorespiration does not occur because they are physically separated. Light reactions, oxygen never comes in contact with rubisco. Cannot fix it. Good. All right, so now we have these other types of plants that are a little different. These are our CAM plants, C-A-M, CAM. Okay, these are things like pineapples, succulents, cacti. How do they survive that heat? Okay, because they are brilliant. Okay, CAM plants have evolved to store CO2 at night. So basically in the daytime, what they do is close their stomata because it's hot and they don't want to lose their water. So they close it. And then at nighttime, they open it. Okay, this is night. This is day. Okay, now we know that tons of O2 is being made during the day. How do we prevent rubisco from fixing oxygen instead of CO2? Well, because the plant is brilliant. So what it does is it opens its stomata at night and it takes in CO2 and stores it away as a C4 inside of the central vacuole. Now, during the day, as oxygen is being made, remember the light reactions can only happen when there's sunlight out. Okay, at nighttime, is there sunlight out? Obviously not. So there's no O2 being made. Now, during the day, while it's being made, the CO2 that was stored overnight is going to be directly fed into the Calvin cycle, blocking this contact between O2 and rubisco, thus preventing the plant from going into photorespiration. And that's that. So, and of course, not only is the light reactions making O2, of course, it's making the ATP and NADPH necessary to run and operate the Calvin cycle. And oxygen will leave. Technically, it's going to leave at nighttime. It'll leave when the plant opens its stomata at night. And it has solved that puzzle. Right, plants are pretty smart, huh? Okay, now let's talk about some pretty important aspects. This is a huge issue right now across the world on our planet. So we're having something called the greenhouse gas effect, global warming, and the reason why is pretty straightforward. All right, I'm going to keep it simple here. It's caused by CO2 buildup, right? Not just CO2, actually. There's other greenhouse gases, like methane gas is one of them. But CO2 is a big player in this issue. And it's causing something called the greenhouse gas effect because we have too much atmospheric CO2. And basically, why do we have too much atmospheric CO2? Well, ever since the Industrial Revolution, where we started mining and burning coal and burning oils, burning fossil fuels, cutting down trees, the planet is in some pretty bad shape here. Now, what greenhouse gases do is they basically create this greenhouse effect. What does a greenhouse do? Okay, a greenhouse is designed to trap heat inside of the greenhouse so that plants don't freeze. Now we have some in California. They're more common in very cold areas for people to have a greenhouse, and we have one at the campus, and the point is the greenhouses trap heat. Now what's happening to our planet is we're having this heat being trapped in our atmosphere. That's why it's called the greenhouse gas effect. So basically what's happening is, okay, sunlight comes in. And this is normal, okay? It's been happening since the beginning of time. It comes in and it's absorbed by our plants, and it's absorbed, by organisms that can absorb the sunlight, and then it radiates back into space. It's supposed to radiate back into space. All right, well, ever since the Industrial Revolution, we have a greenhouse gas effect. We have this layer of greenhouse gases surrounding our whole planet in the atmosphere, okay? And instead of all of this stuff radiating back into space the way that it's supposed to, it's getting trapped in our atmosphere, making the entire overall climate across the globe hotter and hotter and hotter and hotter. Okay, why is that a problem? Well, there's tons of reasons why that's a problem, but the biggest concern is the fact that we have a North Pole and South Pole that's made completely out of ice. Now, what happens if our North and South Pole melt? Where do you think that water's going to go? It's going to go into our oceans. It's going to go into our oceans, and what we know as the shorelines, it's going to get covered. these continents are going to get covered. We're going to have huge floods. We already have floods. This is not, new. It's happening now. So this is a big, big issue. And to top everything off, now if CO2 is radiating around the atmosphere, normally plants pull in CO2, right? They use CO2 to build sugar, and it's all good. Well, you know what? It's not all good anymore because they're cutting down the rainforests, and we're losing our trees that could really actually pull a lot of the CO2 out of the atmosphere because they utilize it. Now, when we're cutting down masses of our forests, well, now we're in trouble. All right, so plants not only make oxygen and food for us, they suck that CO2 out of the atmosphere. And as we cut them down and cut down masses of our forests, we're endangering our planet. Okay, that is the end of chapter 7. Please read your chapter. If you need to watch the video again, please do so, and I'll see you in the next video.